Hey everyone, this is our lesson on meiosis. And by the end of this lesson, you should be able to explain the role of meiosis in sexual reproduction and genetic variation. So, remember, we talked about mitosis prior to this, and that was regular cell reproduction, so asexual reproduction. Meiosis is what your cells go through for sexual reproduction, so to create an offspring. And the goal of meiosis is to produce the sex cells or the gametes used for reproduction. Organisms have tens of thousands of genes that determine individual traits, which are lined up on the chromosomes. And then cell division produces cells with half of the number of chromosomes as the parent organism body cells have. So we know that our regular cells have 46 chromosomes in them, but meiosis, those gamete cells will have 23. This is known as reduction division, and this occurs in the cells that produce your gametes. So for females, this occurs in the ovaries. For males, this occurs in the testes. So we have phases for meiosis, but meiosis is broken up into meiosis 1 and meiosis 2. And the phases are all the same names as the phases in mitosis, just with a 1 or 2 after. So we do still have interphase here. We have a prophase 1, a metaphase 1, an anaphase 1, and a telophase 1. And in the interphase here, this is where the growth and DNA replication occurs. So it is the exact same as what happens in mitosis. Then for prophase 1, you should see all of the first set of like meiosis 1 and mitosis are basically identical. So here we've got the chromosomes will coil and the spindles will form just like normal. The nuclear membrane disappears. The nucleolus disappears. And then the homologous chromosomes pair up and form a tetrad via synapsis. So they're going to pair up just like we saw originally, and that tetrad is going to form. Then this will allow for what's called crossing over, and this is the exchange of DNA segments between homologous pairs. So you would actually see parts from two identical chromosomes, and they will split and cross over and go to the other pair. And this result of this crossing over process is gene combinations on the chromosomes can be changed. So this is really, really useful for genetic variation. So just as an example, and this is in your notes, or should be in your notes, if you have one chromatid of a chromosome initially contain genes for brown hair and brown eyes. After crossing over, it could contain genes for blue eyes and brown hair by taking the gene for blue eyes from one of the chromatids of that chromosome's homologous or homolog. So you can actually change and express different genes in the offspring than in your parents, but they had to come from somewhere. So all of our other 1 phases, so metaphase 1, the tetrads will line up, so you can see that here. Instead of 2, we see 4, okay, that tetrad. Tetra in science means 4. So that lines up on the equator of the cell. And then in anaphase, the homologous chromosomes will separate at that centromere, so they kind of split. One chromosome will move to each pole, and the cells will start to elongate so that they can start that telophase of kind of cinching in the middle. So the spindles break down in telophase 1. The chromosomes will uncoil back into their chromatin. And then the cytoplasm will divide so that you can have those 2 cells. And then each cell will contain 1 chromosome from each pair. So now you have 2 identical cells. Again, just like mitosis started off, except I shouldn't say identical, because if you do have that crossing over occur, they won't be identical anymore. They're going to express different genes, but we will still have the same amount of material in each cell. So then we move on to meiosis 2, which is the second set of the cell cycle. a second division occurs here. So there's no replication in meiosis 2. Okay. So the chromatids of each chromosome will separate. And the goal here is I always struggle with this word, gametogenesis. And this is the formation of your gametes. So this is the formation of the 23 chromosomes versus the 46. And then the gametes, again, remember, are the reproductive cells. So all of these also have your pro, meta, ana, and telophase, but now they're twos. So prophase 2, the spindles will form in both cells produced by meiosis 1. In metaphase, the chromosomes in both cells move to the equator. In anaphase, the centromeres will divide, and the sister chromatids will move to opposite poles. And then in telophase, the nucleus will reappear, will reform. Spindles will disappear. The cytoplasm divides, and this will result in 4 haploid cells. So remember, a diploid has double the chromosomes. Haploid has half the chromosomes. And then remember that each has 1 chromatid of the homologous pair instead of the pair itself. Now we can talk about the 2 different types of reproductive cells and gametes in males and females, and basically how they're formed. So we have spermatogenesis and oogenesis. So spermatogenesis, this will occur in the males, and this will produce the sex cell for males, which is sperm. 4 are produced per meiotic division. And then we have oogenesis, which occurs in the ovaries, the females. Here we have the gametes, which are produced, and we know those are eggs. And then the cytoplasm divides unequally in these. So you have one ootid, which is a cell that receives the most cytoplasm, and you have 3 polar bodies, so little cytoplasm, and these will disintegrate. So these are not viable. So we only get 1 haploid egg for meiosis in females. Now let's talk a little bit about genetic recombination. And here's where you'll see more of that crossing over concept that was mentioned earlier. And the whole point of genetic recombination is to make new or different genes in the offspring. So we don't look identical to our parents. At some points in our lives, we may look more like our mother. We may look more like our father. We may be a perfect mixture of both. And that's all due to this genetic recombination. And there are 2 different ways that this will occur. One of them is independent assortment, and one of them is crossing over. And this is very, very important to genetic variation, which gives offspring a better chance of survival. So the whole point of gene expression is to be able to survive in different environments. This is one way that offspring can do that. And this may allow your offspring to survive in a changing environment. So the first one we're going to look at is independent assortment. And this is where the chromosomes will pair up and line up in 2 different ways. So for humans, there are 8 million possible combinations in eggs and sperm that you can look at for independent assortment. And upon fertilization, the human zygote has 70 trillion possible combinations, just depending on which genes are there and expressed on that karyotype, then will show what the offspring possesses. Then we have crossing over, again, what we talked about. So this is exchanging genetic information between chromosomes. So we're swapping genes on a chromosome. And actually, let me show you, I'm just going to go back really quick. This GIF that you'll see right here, if it will move, I don't know if it will. It may not move again. Yeah. Well, if you saw this GIF earlier, you would have seen how they basically unzip at a part and then re-zip on the other side and then move.